Hi biologist, welcome to your next screencast. Today we're going to continue with chapter 7 and our discussion of DNA structure and replication. DNA profiling is another name for DNA fingerprinting. When we do DNA profiling, we don't want to use a person's entire genome because sequencing it would be extremely time-consuming and expensive. On top of that, humans share between 99 and 99.9% of their DNA. Looking at all of that would be inefficient and ineffective. Instead, we use a shortcut. We look at DNA between people that's different. We use PCR to amplify those specific segments of DNA. These segments are called short tandem repeats, or STRs. STRs are sections on a chromosome that have a DNA sequence repeated. For example, you might have the sequence AGCT repeated over and over. Let's look at an example. The person in the diagram has an STR region on chromosome 7. There are 6 copies of the STR on his maternal chromosome, and there are 4 copies of the STR on his paternal chromosome. The maternal chromosome came from his mother. The paternal chromosome came from his father. STRs are found in the same places along the same chromosomes in different people. The exact length of STRs can vary from person to person and between maternal chromosome and paternal chromosome. You can see this in the diagram. The 3 people here have different STR patterns in the STR region on chromosome 7. These differences can help distinguish one person from another. The first step in making a DNA profile is to collect cells and extract DNA. Cells can come from anywhere in the human body, including saliva, blood, skin, and hair. Next, we'll use PCR to amplify or make many copies of multiple STR regions. The more STR regions we use, the more unique the DNA profile. The last step is to separate STRs by using a lab technique called gel electrophoresis. DNA has a negative charge, and we can use an electric current to separate DNA fragments by size. Smaller DNA fragments travel farther and faster. Larger DNA fragments travel not as far and more slowly. The DNA fragments containing the STRs separate out and form a specific pattern of bands. Remember, shorter fragments travel farther than longer fragments. The pattern of bands created by DNA profiling is unique to each person. We can compare patterns of DNA for a number of different purposes, including proving guilt or innocence or demonstrating a relationship. Different individuals have different DNA banding except identical twins. When we do DNA profiling, we want to use multiple STRs. For every STR you use, you get 2 bands, one from each parent. One band comes from the maternal chromosome, and the other band comes from the paternal chromosome. On top of that, the bands can overlap and appear as one band on your gel or DNA profile. To make sure that no 2 people have the same DNA profile, we want to use a combined pattern of STR repeats at multiple sites. This diagram shows the location of different STR regions on different human chromosomes. It also shows that if you use one STR region, then 1 in 5 people will have the same DNA profile. If you use 15 STR regions, then 1 in several quintillion people will have the same DNA profile. That's 18 zeros. While not being perfect, DNA evidence is more reliable than other forms of evidence. For example, bite marks, when used as evidence, can have an error rate of up to 91%. Hair can only be used to exclude people. It cannot positively identify someone. As I mentioned before, except for identical twins, no two people share the same DNA or the same DNA profile. The FBI has a DNA database called CODIS, which stands for the Combined DNA Index System.